can see everybody.
Dr. Ram, you know anyway. Dr.
Yeah.
You have talked, I think.
Okay.
The one person which is seeing new is Dr. Arvind.
Yeah.
Right? I think you have talked to him already.
No, he's looking
He's
at
a
the
Dr.
screen.
Bangladesh.
Yeah, he's looking at
Hello, sir.
Hello.
Good morning.
Good morning.
Morning.
So, madam, you can start.
Yes, sir. As you said. Namaskar, Suprabhatam to everyone. This is Namrata Bajaj from Indrashil
Ma'am,
University.
just 1 second, ma'am. Just one
Yeah,
second.
sure,
Just
sure.
1 second. Just 2 minutes. We can wait for the attendees to join.
Okay, fine. And if the PPT is ready,
Yes, ma'am. Just 1 second, ma'am.
yeah.
You know, participant figure
It is crossed 500.
when cross 500, but total. You know, Arvind,
Yeah.
it had crossed more than 500 registration.
Oh my God.
Let us see how many
Yeah.
will attend. Already 162. So they are attending. So we'll just wait for.
Yeah, there is just one little problem, sir, that one of the link is down, and I'm using another link. So in case it breaks down, then it will be difficult. Then we'll have to probably stop in between, and I'll have to find out how to connect it. Hopefully that will not happen, because we usually have two links, but one is completely down from the morning.
Okay, okay, okay.
But what you can do is you can share your presentation with Dr. Yadav in advance, and then you can join by mobile phone, and that will work.
Okay.
He has shared his presentation is there, right?
Yes, yes. I already did that.
Yeah.
His presentation is there already. Actually, his presentation, Dr. Ram's presentation is there already.
Anuj, just give the signal to start.
Because already close to 200 are there already. So it's a good number. Because meantime people will join. Most probably they will join at 11 when Arvind lecture comes.
No, sir, already 198 joined on Zoom. On Facebook, there may be many.
Many, okay. 200.
Yeah, 200.
Already 200 joined.
Your face is not clear. What I can just come
But this is not, this is okay? That's it. So, Namrata, we can start, no?
Yes, sir. Okay. Once again, Namaskar, Suprabhatam to everyone. This is Namrata Bajaj from Indrashil University. In the first place, Sadar Abhinandan, welcome to our distinguished experts and panelists who are shining stars of Indian science and have acquired fame globally. Dr. Arvind Sahu, Dr. Ram Vishwakarma, Dr. Bakulesh Kamar, and our mentor and provost, Dr. J.S. Yadav, who is once again a stalwart scientist in chemistry. I also welcome our viewers and participants who are with us on Zoom and Facebook. Please, presentation start करेंगे. Anuj sir. Actually, India has been a land of saints and scientists from very long time. And this is what we see is reflecting in Kedla Pharma as well as Indrashil University is providing a right forum. Shall we start the presentation, please? The present scenario of global pandemic COVID-19 has made everyone realize significance of science, research, and technology as savior of mankind. And Indrashil University, an educational initiative of Kedla Pharmaceuticals, have been proactive pertaining to science to service of society since its inception, which is evident through series of webinars hosted of late, and today we are again having presence of eminent speakers with similar noble purpose. Before we begin, I wish to take you through a quick glance on Indrashil University via PowerPoint presentation. Presentation, please. Yeah.
Ma'am, there is some technical glitch.
Okay, no issues. I'll continue. Indrashil University was established in 2018 as a philanthropic initiative of its founding chancellor, Dr. Rajiv Modi, in the memory of his parents, Indravardhan Modi and Srimati Sheila Ben Modi. They had laid the foundation stone of Kedla Pharmaceuticals in the year 1951. And since last 68 years, it is continuously serving to the mankind in preparing and manufacturing affordable medicines. The founder chairman was a very humble person with very visionary insight. His contribution in pharma sector for affordable medicine to the common man fetched him the title of Medicine Man of India. And similar ethos and principles are observed at Indrashil University also. Next slide, please. See here, this is the graph where in 1951, Kerala Pharmaceutical came into existence, and then it went on establishing Sri Saraswati Education. Indrashil University was initially formed with an society of Sri Education Saraswati. In 2018, Indrashil University came into existence, and in 2019, in a short span of 1 year, we came to AIC, Atal Incubation Center, with the help of Niti Aayog. And then in 2020, we have come up with Indrashil Innovative Foundation also. Next slide, please. We would wish to introduce our mentor and provost, Dr. J.S. Yadav, and the credentials are too huge. It's very difficult for me to give you a brief introduction also, because he is a stalwart in organic chemistry. Thank you. Please, next slide. Next slide, please. We see that we offer at Indrashil University, we offer 2 programs as of now. One is in School of Engineering, the other in School of Science, right from graduate to postgraduate programs and PhD programs as well. Next slide, please. We have all state-of-art infrastructure and other lab facilities to match with the vision and mission of our university. Next slide, please. We are having a curriculum wherein we have a focus activity with MOUs with so many national research agencies, as well as we have MOUs and collaboration with foreign universities. Next slide, please. These are the foreign universities with which we have made the collaboration. Next slide, please. This is what we are offering, dual degree, which has become a trend today. That is BTech in Computer Science and Engineering in collaboration with Sacred Heart University, USA. Next slide, please. These are the industry and academia linkages we have, wherein we are supported with the industry visits, with the industry experts coming on the campus. We also take our students to the industry and get exposure to them, as well as for placement purpose also. We have these tie-ups, and we are helping a very trendy and experiential learning to our students. Next slide, please. This is the most flagship program of our university, that is Atal Incubation Center, and it started in 2019, and till date over 5,000 ideas have been outreached through seminars, workshops, trainings and webinars, and we have received over 300 plus applications till date in this center. And again, the applications screened off above 50, and the shortlisted for incubation support are 30. The number of pre-incubates are 12, and the startups which we have supported are 5. The current campaign which is going on is ISC Ideathon 2020. Next slide, please. This is what we wanted to show the glimpse of Indrashil University. For any clarification or any details, you can contact us at www.indrashiluniversity.edu.in, and the email is also mentioned over here, and you can call at 6359102727. And before we begin, just brief of the norms. We will take all the questions at the end of the sessions when the speakers are through with their session. Then we will take up the questions. The questions can be raised in the chat and the question answer box. Again, you please identify yourself and to the speaker's name also, so that we can answer you adequately. And further, we encourage you to send us any other feedback also. E-certificate is provided to everyone whosoever has registered through proper channel. As well, we will be sharing with you a feedback at the end of the seminar within 30 minutes, wherein you can send your feedback, based on which you will be eligible for getting the e-certificate. Thank you so much. Now I request our provost, Dr. Yadav, for the opening remarks and brief introduction of our esteemed speakers. Dr. Yadav, please.
Okay, thank you, Namrata. We have only 3 minutes left for starting our webinar. The vision and mission of Indrashil University is centered on the service to society and mankind at large using platform of education. IU has hosted several webinars to create awareness and address the challenging issue of the wide benefit of the humankind getting solution through scientists and industry experts. These days, our country and in fact entire world is reeling through a very tough phase of pandemic, COVID-19. The webinar today has distinguished speaker Dr. Arvind Sahu and Ram Vishwakarma, along with Bapulesh Kamar, who has worked in this area for quite some time. We always take in the webinar our current challenges of our society in India, and we have organized one of this is like COVID, which everybody is talking. So what we have taken is 2 people. One is doing the testing of the COVID-19, other is developing vaccine for the COVID-19, rather repurpose vaccine for the COVID-19, which is already having, they will speak, I do not want to speak. Only thing I can say that biodata I'm not going to read, it is already there in the invitation. Only I can say that like Arvind Sahu, he has created facility for testing COVID-19, kind of care it has to be taken and kind of facility it has to be taken with of his own he has taken without caring for that he may also get corona. Without caring for that, he started a social service for the people. And Pune is epicenter and he has that facility, one of the best facility he has, and he's servicing the COVID-19. And same is Dr. Vishwakarma. He was the director of IIIM and retired recently, and he offered himself to continue the clinical trial of CSIR for repurpose vaccine for the COVID. You will hear more about that from Dr. Ram. So I don't want to take much time, already it is 11 now. Now I request Arvind to start his lecture. Okay.
Thank you, sir. I invite Dr. Arvind Sahu, without much ado, to have his views on COVID-19 current understanding.
Thank you very much, Professor Yadav, for asking me to deliver this talk on COVID-19. COVID-19 or coronavirus research is not really my direct field. My direct field is more innate immunity and viruses and how they crosstalk. But like all of us, are kind of driven into this, and therefore we also started COVID, testing facility. Our director, Dr. Manoj Bhatt, was very kind. He had this idea we should start, and since I am sort of resident virologist in the institute, we got all the help from our colleagues who jumped into this, and we started. We also started programs like making neutralizing monoclonal antibody, which is an immediately funded project, and looking at how the coronavirus is changing, particularly in our area, Pune and nearby area, by sequencing whole genome. And Yogesh Sauche, who is the senior scientist, is leading that program. And we have various other little programs to look into the research at NCCS. So what I'm going to do primarily is give the overall impression about what we have learned in last 6 months, because this is a new disease and we are still learning. And there are many unknowns, which you will hear in other talks as well. But what I'm going to do is provide a gist overall and not really get into the details of scientific details of this to really tell you. So can I have my presentation, please? So once we have the presentation, I'll start.
It is important to mention here before it starts, it is important to mention that Dr. Arvind Sahu is doing selfless service, just as the saints have been doing in the ancient India, that they were more of researchers. So is the case here that these scientists have become saviors for Indians, and especially this in difficult phase of COVID-19. It's very difficult, and people are eyeing on all these scientists who are actually saints, who are trying to save human lives. So really we are thankful to all the saints, scientists, doctors, who are actually tirelessly working on this pandemic, that how to curb and how to contain that. The technical team is requested to please sort out this.
Time is going.
Yeah, I'm really thankful to all my colleagues who jumped into this and working, tirelessly, 7 days a week, 24/7. This is quite laborious. I didn't realize it, that how much time it really takes, right from collection till the reporting. And our life starts early in the morning and we finish, by 10:30. And this is like 24/7 jobs. And then we have to report, keep talking to people, arrange the samples. So we have a whole team of my colleagues who are looking into different aspects. And that's how we are able to manage this.
Actually, people aren't aware that our scientists are doing, this kind of laborious work, and still people are speculating that what India is doing, what Indian government is doing. But actually, behind the curtains, things are going on very rigorously, and we are aware that. And this is why Indrashil University has hosted this webinar to sensitize people that see how our scientists are working day and night tirelessly to save mankind, and how we can use science and technology for the survival of human life, to make their life easier. I think we are in position to overcome that technical glitch.
अभी तक तो नहीं आया हो. Okay. Meantime, I did mention about Dr. Bakules Kumar from Cadila Pharma.
So, sir, should I try to share the screen from my side?
Okay.
Yeah, I'll just check with my technical team. Is it possible to make him host and allow him to share? Yes, sir.
Okay.
So I am sharing from my side. Can you see it?
Yes, sir.
I can see, right.
Yes, sir.
Okay. Okay.
Please go ahead, Dr. Sahu.
Okay.
It's visible.
Yes, sir. It is visible.
Okay. So, I changed the slide. Can you see that the slide is changed?
Yes, we can see.
Yeah. Okay. So, basically I'm going to summarize about COVID-19. So the coronavirus disease, 2019, and what we call shortly as a COVID-19, we all know that is caused by a novel coronavirus, which we now named as SARS-CoV-2, because there was already SARS-CoV-1 earlier, which disappeared mysteriously. So what the coronavirus, the first question is why the name coronavirus? And in Latin, the word corona means actually the crown. And since these viruses have spikes which look like spikes in the crown, they are called coronavirus, and that's how the structurally they are defined. You can see the structure which is shown here, the EM structure, where you can clearly see the spikes. So coronaviruses basically are zoonotic viruses. What it means basically that these are there in the animals and they jump the species and they start infecting humans. So this has been happened earlier. And the peculiar thing about coronavirus, that it causes respiratory distress, and this particular one causes really, a lot of problem all over the globe. The symptoms what we get, as we all know now, very well publicized in the media, that the symptoms are pretty much like flu. So we get fever, headache, muscle ache, cough, sore throat, and breathlessness. So these are the common symptoms. There are many more other symptoms which you see in severe patients, but these are the normal common symptoms which we see. So where did it all start? So this started in Wuhan, in something called the Hunan fish market, or Hunan wholesale fish market. I think this is a misnomer, the name fish market, because I think it seems that pretty much everything that moves is sold in this place. And you can see that there very little section, one side is a fish, but you have everything else which is also sold, including bats and everything, you can name it. So the whole story really started here. So let's look at the timeline and the origin and the spread of the virus. So the first case of this mysterious coronavirus, or SARS-CoV-2, actually happened in December 8. And on December 31st, the China alerted the WHO about this mysterious pneumonia-like disease. And that's when the whole world came to know that something is going on in Wuhan and there is some mysterious disease. Later on, this was not just limited to Wuhan, it moved out of the China, and the case was reported from in the Thailand, and on Jan 13th. Now, after this was reported, and getting again and again, WHO immediately declared this as a global public health emergency on January 30th, and that's when we also in India got the first case in Kerala. Now they started actually moving into different countries, and more cases started, in countries started reporting. And this was almost reported in 100 countries with over 100,000 cases, and on March 11th, then WHO declared this as a, and that now this is global, this is everywhere, and this is becoming pandemic, and they declared it as a pandemic. So as of 20th July, we have over 14 million cases in 188 different countries, over 500,000 deaths. But the good thing is that a lot of people are recovering. So there are recoveries almost more than close to 8 million people, which is a good sign. So let's look at the timeline in India. So as I said, it got first introduced on Jan 30th in Kerala. On March 2nd, we also had cases in Delhi and Hyderabad. And then slowly, this started more cases, and we then, our Prime Minister declared Janta curfew on March 22nd and a complete shutdown. for 21 days, which was declared by the Honorable Prime Minister on March 24th, which went on till the end of May actually. And we then started opening up slowly. And currently we have over 1 million cases, and out of which 4 lakhs are still active. We have good recovery, about 7 lakh people recovered, but unfortunately more than about 27,000 people died during this outbreak. So as you know, of 20th, if you see, we have many confirmed cases, and a lot of cases now. Maharashtra has become a hub. So yes, we have a lot in Maharashtra, also in Delhi and Tamil Nadu, and now of course Bangalore and in Karnataka is also increasing. So there are many active cases, close to 4 lakhs now, but good thing is that the recovery rate is pretty good, and about 63%, and over 7 lakh people have recovered. So the question obviously is, why this disease really became pandemic. I mean, coronaviruses are there, they are zoonotic viruses, they do jump, and they have been jumping, and we know earlier MERS and SARS-CoV-1, and also there are seasonal coronaviruses which you see in Hong Kong. So why this? What was so peculiar about this particular virus that it became pandemic? So I tried to jot down few things. So the first and primary reason obviously is this is a novel virus. We were not exposed to this particular virus, and our body did not have any pre-existing defense against this particular virus. And therefore, once we get exposed, there is nothing which we have pre-existing immunity, and therefore it starts flaring up. The second important reason is it's airborne. So people who are carrying, and when they are traveling from one place to another place, and particularly now world has become a global village. So people are traveling across the globe and they're carrying this virus, so it's very easy to spread. And the respiratory viral diseases are one thing which spread. And last time, we came across H1N1, which got, everywhere. And now we have the coronavirus. So respiratory viruses are very easy to spread, and this belongs to that particular category. The third thing is it has very high transmission rate. It transmits very easily anywhere. One person can give it to around 3 people is what we has been estimated. So the transmission rate is again very good, which also makes it that it can spread very easily. The fourth point is long incubation period. People when they are infected, it can be anywhere from 1 to 12.5 days. And so they are incubating, they are traveling, and they don't know that they have this infection. And therefore, they are carrying to different places. And that's how the virus can move from one to another place. The another problem, obviously, is asymptomatic transmission. We know that many people have asymptomatic cases, they have the virus. Again, in this particular cases, we have seen during, when we are testing that the load is pretty high. It is not that asymptomatic people carry less load of the virus. The load is pretty high actually in some, in many cases, much more than even people who are actually symptomatic. So they are carrying and they are sort of, giving the virus to other people. So that's another. So together, this has made it a pandemic. So these are the main points which I have come across. And if you compare the SARS-CoV-1 versus SARS-CoV-2, you can see that the SARS-CoV-1 was not, it was more pathogenic. This one is less compared to SARS-CoV-1, but you can see the number of cases, there is a steep rise, and this went everywhere very fast. So let's look at the numbers, particularly for the SARS-CoV-2. We all know that when we talk, when we cough, when we sneeze, the virus is liberated in pretty high concentration, right? So even when we're talking, we are actually giving away the virus. But is there any other route where the viral load is very high and the stool, uh, the load is again very high? So, what does it really mean that when we talk, uh, and how many particles are really needed to get infected? And as low as 1,000 SARS-CoV, people believe, and it hasn't been estimated that scientists, they are enough to make, infect. And what it means to translate that if you are talking to a person who is, infected for 10 minutes, then you can get this infection. Okay, face to face, if you're talking to a person for 10 minutes, you can get this infection. Scientists also looked at the environmental stability of this virus, that now we know, it is can go through aerosol also, which means that when the size is less than about 5 micron, then they are not called as droplets, more like aerosols, smaller ones. And they can hang on in air for almost 3 hours, right? So if it is a closed rooms, then they can hang on. If it is an open area, obviously, it will get diluted. So the roughly half-life in the aerosol of this virus is about 3 hours, which was published in New England Journal of Medicine. What about the surface? We always say that surface are contaminated, one should not touch it. How long they really stay? So if you're talking about plastic or the stainless steel surface, then they can hang on there for almost 72 hours. They are infectious for that long. Copper somehow, they are not so good. So they don't hang on so long. In copper, only 4 hours. But cardboards and stuff like that, they can stay almost a day, 24 hours. Okay, so they are there for quite a while when it comes to the surface, almost up to 72 hours. So that is something, the number one needs to keep in mind. So what about the virus? How does it look? As I said, the name coronavirus itself means that it has, spikes, crown-like spikes. So these spikes are basically trimers. They are trimers of spike protein. And the peculiar thing about this, that the affinity in this particular virus, the SARS-CoV-2, is very high. It's in nanomolar, anywhere from about 40 nanomolar, which is much higher than the earlier SARS-CoV-1. So this is, one of the reasons why, it infects so well. Another thing is, the question is how big the size of the particle is. It's around 100 to about 120 nanometer in size. So that's something like 1/100th of a hair, thickness of the hair. So that's the size roughly of the cell, which is pretty big, but it is almost same as what one sees like a flu virus. The size is pretty much same. If one compares, the genome of this and looks into what about the other coronaviruses like SARS-CoV-1 or MERS or the common cold, then what you see is that it is more closer to the bat CoV, but it's still not, same. It's only 96%. What it means that what is predicted is that when it jumped from bat to human, there was an intermediate species. So it went into some intermediate species, and from there it jumped into human. What that intermediate species, we still don't know, and people are still looking into that. So is virus changing? And that's something one would like to know. So this is a RNA virus, right? And RNA viruses are known to mutate 1,000,000 times higher than the host. So they mutate a lot. So what about the coronavirus? Is it mutating? It is mutating. It has a proofreading activity, so the mutation rates are a little lower, but it is mutating and the rates are lower. It's about 4 times lower than the flu virus, which we know very well. So the mutations are there, but the rate is not that high. but it is mutating, and almost in different places it is mutating. So one of the theory which people, always think and discuss and talk is that, when the virus comes and stays for a long time, it becomes less and less pathogenic. It gets mutated and becomes less pathogenic. So far, we have not seen that the virus SARS-CoV-2 is getting less pathogenic. The mutations, which are particularly in RDRP and S protein, and these are linked mutation. There are 4 nucleotide variations which the world has seen, and these are linked. And 2 are the ones which change amino acid. One is in RDRP and another is in the S protein. And the S protein change is at the 614, which is quite talked nowadays about. If anything, this has made it little more pathogenic. The transmission people have seen is more, and the infection rate is more when you get, this mutation from ASP to GLI. And so what we see overall, that the pathogenicity so far doesn't seem that it is going down. So how do we test that a particular person is exposed or infected with the virus? The most common is directly looking at the virus, and what we look directly primarily is looking at the viral DNA by the RT-PCR test, and pretty much everyone now knows about the RT-PCR test, where we use different probes and we try to see if these genes are there, and if we see, more than 2 genes, so we initially look for each gene. We see more genes, we say that it is positive. The other test, which is more to do with the point of care test, is the antigen test, where we directly look for the antigen. Now, this one is not as sensitive as the RT-PCR test, but if the load is higher, particularly in SARI cases, people see that always they will do antigen and it comes out positive, as what has been told to me by physicians who are directly dealing with these patients. So antigen positivity test is also another test, which is more like a point of care test. But if it is negative, then still one needs to go for doing RT-PCR to make sure that the person is negative. The third one is the antibody test, much talked about. This doesn't really tell that the virus is there or not there, but what it tells you is that the person has been exposed to this particular virus. There is a prior exposure. Whether it's active infection, it's not, you cannot say based on the antibody, but what you can say that and this is more for, getting, you want to know what population, what percent of population is getting infected is pretty good way of looking at it. And that is what India is also doing. So we all know that, most cases of these are asymptomatic, or they have very mild infection, which means that the people don't even know that they are actually suffering from COVID. And this is almost 80%, which is a very good scenario. The only problem is that since these people don't know, they can transmit it very easily to other people. The remaining 20% by and large require hospitalization, and they have to see the doctor, they have to be there, and then there are multiple. And 10% of this, they become really sick, and they need various support. And some percent, anywhere from 1 to 3%, die from this, and they become really, really sick. And in many cases, and people, we do understand a little bit about what is causing the severity, but there are still unknowns which we don't really understand. So the mild symptoms, everyone knows what the mild symptoms are, right? And when the symptoms become, more shortness of breath, you have the oxygen level started dropping down, and one needs to worry about it. So fever, if you have a fever, high fever, and shortness of breath, you must see a doctor. And it has been told again and again that one should look for the oxygen levels, and see if it is dropping more than 95. And it's critical when it's less than 90, but one Physicians say that if you're dropping less than 95, one should immediately be alarmed and see the doctor. So what is happening in severe cases? So once we get viral infection, our body tries to protect us, from the infection. So basically the innate immunity, which is much talked about, gets kicked in. And this is a non-specific immunity, which we try and, tries to protect us. And the specific immunity comes into play around say day 10 and after that. And, during this process, various cytokines and chemokines are produced, which are actually there to help our body to make this specific immune response. But what happens that sometimes they become very aggressive and they start destroying. So one of the reason in severe cases that something called cytokine storm, which is there, which is generated, and that is actually making it more severe cases. So this has been reported very well in the literature. So what are the treatments? Mild cases, pretty much everyone knows hydroxychloroquine is something which has been said that, and is been given. Favipiravir is also given, and these are the mild cases. So by and large, you treat. In moderate cases, the problems are becoming more. Remdesivir is suggested, hydroxychloroquine or the combination of both, but also the steroids. And we know that, there are small clots are getting formed in patients, which has been reported. Initially, we didn't know about this in this disease, but later on people found. So anticoagulation and also the convalescent plasma, and much talked about now, is something which is given in moderate. In severe cases, I said like cytokine storm is there, right? So the monoclonal antibody against the IL-6, something called itolizumab, very difficult to pronounce. I don't know why people give these kind of names which are difficult to pronounce. I don't think even the B cell people like this kind of names. But anyway, itolizumab, if you don't remember, just is the antibody against the IL-6 receptor, which is there to block the cytokine storms. Other things which are given, say, steroids, anticoagulation, treatment, and so on and so forth. And these are some things which are there. Of course, it depends on the condition of the patients and what symptoms are, and the physicians take a call based on that. I'm not an expert in this, but I just wanted to introduce is how the treatment is done. So the bigger question is it here to stay? And the short answer is yes. So initially people talked about that maybe there is a herd immunity and, we'll get the herd immunity and therefore will be protected. And this, it turns out that, one person can give it to 3 people. And if you do the math, about 66% of the population must be exposed to this. And that's a lot. And if we wait for that, a lot of fatalities will be there. So the question then is, how this is going to be contained, or will it be contained? Or basically, what about the other coronavirus? What did we hear, right? And what do we learn from those? So, and are they still there? So for example, the MERS was another coronavirus, which is much, much more fatal than the SARS-CoV-2. And this is, there is still limited circulation for this. SARS-CoV-1 somehow disappeared mysteriously, and we don't understand how that disappeared. And there are also seasonal coronaviruses like flu, which happen every day, something like Hong Kong. And these are there. So what are the factors really, which determine the fate of any virus? One obviously is the environmental effect on the viral stability. Is it stable? How stable? This looks pretty stable. Transmission rate, this is very high again in this case. Host behavior, how do we behave? How do we try and contain this virus? What precautions we are taking will determine a lot on this. And changes in the immunity level over time, either by herd immunity or more importantly by vaccination. And that's what is going to decide, whether in how long this is going to stay. But my gut feeling is that this will become more like a seasonal infection, what is there about seasonal coronavirus, what we see in Hong Kong, probably more like that is what is going to happen. So will it be contained? India obviously sees a lot steep rise. If you see from the graph on your left, the something which is going up is India. But the good news is that it did get contained in other places. For example, Spain, Germany, Italy, uh, these places, if you see, there is a plateau. So there is a peak and followed by plateau, and we are still haven't reached the peak. So unless we reach the peak, and then it will start going down, but we haven't reached, which means that we are still, increasing in this. So what is going to help? More testing, more isolation, and more treatment. But it is going to get contained. It did happen in other places, and there are many countries which doesn't have, which have been declared as COVID-free, something like New Zealand and other places, right? So it's pretty good news that it will be getting contained soon. The last I want to talk about is the vaccines, which of course, the best thing to, anything which we would like to contain with any viral infection are the drugs. Drugs are always much helpful, but in this particular case, we don't have good drugs, and therefore vaccine is the only hope what we have. But the good thing is that many efforts are being made. There are over 50 vaccine trials which are going on currently. The most talked about obviously is the Oxford vaccine and something called Moderna vaccine. And we have seen the data on the Moderna vaccine, very good response, which you can see on your right side, that good response after the giving 25 microgram as well, a single injection, you get very good neutralization titer, almost as good as convalescent. We have also seen Lancet. Everybody was waiting for the Oxford data, just published, a day for yesterday in the Lancet, and again, very encouraging results, good response, both antibody as well as T cell response. There was also a paper in the same issue of Lancet from the Chinese vaccine. They are also trying. There are different vaccines they are trying in China, right? And our own biotech company, Bharat Biotech, has a Covaxin, which is kill vaccine, which is also trial is going on. So various trials are going on and good encouraging results. So it's very positive feeling at present that something is going to work in this area, but we need to wait and see until we see the challenge studies, which I don't know how they will plan, but obviously the phase 3 data one needs to see. But the results so far are very encouraging. So I would like to end my talk here. Thank you very much for listening to me. Do wear face mask. And 5 points to remember, avoid gathering, be brave and don't panic, clean, disinfect, and most importantly, educate yourself. Thank you. Thank you very much.
Thank you, Dr. Arvind. So you said you have some engagement. Question
Yes.
we'll take now, or whether you can come back for 5 minutes around 12.
We can take questions
Arvind,
right
sir?
now. Yeah.
Yes, sir. Yeah, thank you so much, sir, for sharing this wonderful information. And especially, am I audible, sir?
Yes, you are audible.
Yeah. And secondly, if you permit, we can take quickly 2 questions, because I heard that you may be leaving, or maybe you can answer these questions once you are back, whichever.
No, I can take a few questions.
Sir. One question is,
go ahead, please.
Any specific reason to 0 cases in country near China, like Laos, Thailand, Vietnam? That is what the question is raised.
So see, it all depends on how good you do the testing, and after that, how do you isolate and how do you treat? So these are the 3 points. So we don't have vaccine yet, and therefore the only way one can contain is do this, testing, as it is said time and again. One needs to test. One need to test enough, so that most population, when you are testing, it is almost like 5% positivity. You need to really hype up the testing, which is kind of difficult. It's not easy to, increase the testing. So there is a limitation because, automated testing machines are not available everywhere in India, which can ramp up really testing. We are doing manually. So there are difficulties associated with, but the only way to do it is, test, and then of course, isolate them so that they do the, and after isolation, again, treat, because most cases are asymptomatic. And if we are unable to find out who the asymptomatics are, they are going to spread the infection.
Thank you, sir. The next question is, how many phases are required for a vaccine to be called as successful?
So ultimate success is when you see protection, right? So, how one should see that? So if you have immune response, which is good, which is specific, and which you have the neutralizing response, which means the, antibodies which are raised, can neutralize the virus. What you have is the specific T cell response, which is good, protective. But these are all in vitro, what we are trying to find out. What is most important is that if the person get exposed after the vaccine, is that person protected or not protected, right? So there have been talks about that maybe one should do the challenge studies, which means that give the vaccine to young people and then challenge them. This is very difficult to do, and there are ethical issues associated with that. The another way is you find area where you have a lot of cases, and then you find people who are not exposed, and give them, and then let them stay there. They get exposed equally, and then find it out really who is protected and not protected. And until we have that, that is basically the phase 3 data. When you have, we will know whether the protection is there or not there.
Thank you, sir. Another question is from Chetan Bhausar, who says that what are the chances to get infected again once the COVID patient has recovered?
Okay, so initially the belief was there that once you are having a disease, the response will be very good. You will get very good immunity, and you will be protected. But what people have seen is that this is not happening in all the cases. And in certain percentage of cases, it seems that the immunity boost up is not as much as what you see. And this is something, which suggests that there are chances of getting reinfected, right? So if you have getting infection, the chances are definitely low of getting reinfected, but that doesn't mean that the chances are nil. And that is what I would like to say.
So, but
Okay,
when we
Namrata,
talk
we'll
about
have the last
antibodies,
question because we are running out of time. Okay.
Yeah.
yes, sir. If you permit one last question, the question is how long the antibodies last in a recovered patient?
So that varies, depending on the antigen and depending on the vaccination strategy. A lot of factors depend, what kind of antigen is there, what kind of adjuvants you are giving, and so on and so forth. So ultimately, once you give and the immune response is there, again, the lasting will vary. We don't have good data on that at this point of time. And so it's very difficult for anyone to answer that whether it's going to have a long lasting. Another complicated thing is that these are RNA viruses, and therefore if they change, then that becomes another problem. So what we, for example, if you compare that with the flu, in flu we have seasonal vaccines, right? So every year we have to get a new vaccine because the virus gets changed. But something positive about this virus, is that it doesn't mutate as much as the flu is mutating. So if we get pretty good immune response, it's good chances that, in this whole season we'll be protected. So let's wait for the vaccine results, and let's hope that it will be all good.
Yes, sir. Thank you so much. You have given a lot of hope to us, Yeah, a lot of hope has been given by him, sir. And especially he has brought smile when he says that there is a possibility that it may change into a seasonal kind of virus, and may disappear altogether. That is what the hope we can live with. And I, give charge to our provost to say something on this.
Okay, Dr. Arvind, very nice presentation. and I think it was a self-explanatory everything. And I thank you really for a very short notice. You agreed and adjusted your time from your meeting. And I'm sure we will meet again or some occasion.
Thank you very much, sir, and
Thank
thank
you so
you
much, sir.
for asking me to give. It's a pleasure. Thank you very much.
Now our next speaker is Dr. Ram. He was a director of Indian Institute of Integrative Medicine at Jorhat. He just retired and currently he is a CSIR advisor and he is the one who is doing clinical
Dr.
trials.
Yadav, you have sent me from Jammu to Jorhat.
Sorry, Jammu, Jorhat. I was talking about Jorhat.
Very long journey.
Okay, sorry, Jammu. IIIM Jammu. Sorry for that. And he worked on vitamin B12 by some very tough problem at Cambridge University. Then he was in the industry like Nicholas Piramal. Then he was in National Institute of Immunology, Delhi. And then he was, working with us for quite some time. And he has done a lot of work on medicinal chemistry. And it's specific to mention here that his association with Cadila Pharmaceutical in clinical trial of Sepsivac to mitigate COVID-19 with all aims in Institute of Medical Science, BHU, and other institutes. And it reflects his dedication to the welfare of mankind that he has offered himself to do this job. And I'm sure you will listen more and then you will know what kind of work he is doing in this area. Dr. Ram, it is for you.
Thank you, Dr. Yadav. If somebody can load my presentation. Yeah. So I think you heard the previous speaker giving you about the knowledge about the virus, how it came, how it spread, what's likely to happen. What I will going to tell you what actually we have done on this virus. When the virus was kind of COVID-19 was detected in December, it took almost 1 month to figure out what is going on. And then we had a meeting of directors meeting in Goa in the month of end of January. And I think we quickly realized that we have some serious problem in our hand. And being a very diverse organization with CSIR is, we decided that we will have multi-pronged strategy to deal with this problem. And times like this, people realize the value of what I call investment in science. Okay. Of late, you must have been hearing in last 10, 15 years that people in New Delhi keep asking scientists, what is return on investment? Okay. All those people who are talking return on investment now hiding and looking for scientists and science to deliver something. Every day I get phone call from top politicians, ministers, and they are all afraid to die. And same ministers and politicians, when we went to funding, they said, what you people are doing. So I think this is the time that we make you realize that country like ours, despite being a very poor country in 1947, continued to make investment in science, and our early politicians used to call it investment in future. And that is how these wonderful laboratories system was set up. So I think times like this, people ponder that why it is important to have a consistent, regular investment in creating knowledge, because the greatest and the most powerful country is right now on the knees. You can see that with all the resources available to them, all the militaries and power. So I think this is the understanding we should have, that science is something, and I believe that science will find solution to this problem also. And with this, so we decided in CSIR that how to deal with this, and we kind of divided ourselves in 5 different verticals, okay, because of the strength of the organization. And we decided that a strategy group will be created, made up of 8 directors. And we have been meeting for last 4 months every day from 6 p.m. to 9 p.m. the DG of CSIR and all 7 directors. And I have been coordinating all this effort. So my role is basically to driving all the programs that are happening in CSIR. So that is how we have been monitoring very closely that what should be done. So we divided into 5 verticals. The first vertical is basically molecular and digital surveillance, about the knowledge about the virus, how do we do the surveillance? And now we are realizing, I think in this month, that how important surveillance is. So I'll give you a little brief background of what we have done in surveillance. The second vertical is basically testing and developing new diagnostic kits, both from the viral side and from the host side. The third major vertical is therapeutics and vaccines effort. So I'll give you some snapshot what we have been doing. And fourth vertical, what very important, was the medical devices and PPEs. Because now our situation is better. If you remember month of February or January, there was serious shortage of PPEs and ventilators and all these devices in this country. And we realized that, how dependent we are on the outside world for these devices. And doctors and medical staff, nurses, they were frontline people were exposed. So that area we decided. And the fifth area that we decided was to deal with supply chain. So whatever you want to do in the time of crisis, I'm sure people who works in a pharmaceutical company and corporation will understand the value of supply chain, which in public system we never realize. We take everything for granted that somebody taxpayer will pay for us and we will get it whatever we want to do, right? So supply chain management in terms of creating programs was a major issue. So we decided that, and using new IT platforms, how do we deal with the supply chain, or particularly, for example, you want to manufacture a particular drug or vaccine. So I'll give you a little snapshot of that. So if you can go to the first slide, next slide. Yeah, so I think this, my previous speaker may have covered, but I will just tell you that what were kind of challenges that we had at that time. I'm talking about now the first month. We had very limited diagnostic capability and capacity, no point of care diagnostic, no vaccine, no therapeutic, and we had a very highly infectious virus. This virus is much more infective than its previous sister, which is CoV-1, which came in 2003. And this virus has some special characteristic which no other previous virus had it. So there is something we are dealing with. We should not just deal with that. We are dealing with another virus. It will become seasonal, all this wishful thinking. Let me tell you this. This virus is a very different kind of virus. Its biology is very different. Immunology is different. And every day we are learning, like today itself, the paper has been published in a top journal that it says that very fast we are losing the creation of body to create antibodies. Within 1 month, we are losing when you challenge it. So anybody who's hoping that some vaccines will come and all this, I will temper that expectation. It has been 40 years since HIV came and we have 0 vaccine at this stage. So developing vaccines for virus is not as easy as it is. There are other approaches that we have to take, like, for example, dealing with the immune system. How do we deal with Th1, Th2 responses? How do we deal with other situations? That's a much more effective strategy. And of course, therapeutic. So hopefully we have moved across. And of course, now we are realizing very quickly that it is not one disease. There are 2 diseases that we are dealing with. The first disease, which is for only 1 week, 7 days, first 7 days, when virus is multiplying. That's one part that has to be dealt with differently. Then there is a second week when virus has nothing to do with it. Your immune system is destroying your organs, basically. And some of the data that is available to us is pretty frightening data. We recently had a discussion with King's College team on discussing Dr. Mauro Gica. He used to be Director General of ICGB. And some of the data that he showed us from the autopsies of the dead people, that the lungs have been actually virtually destroyed. So whatever lung capacity that you lose because of this infection, even if you recover, you have lost that part of the lung forever, for your life. Or the brain part that you have, like, for example, a stroke. So I think there are serious manifestations, that a lot of damage that is happening, which is irreversible to the organ. So we should take it very seriously. And that is why it's very important that we take all the precautions that we take. Don't take it lightly that, another seasonal virus and all that. That's not the situation. We are dealing with something very, very different. So here in this vertical, I think what our strength was, and that is how we started working on it. I think this part has already been covered with this virus came and all that. That doesn't matter anymore. That's a more academic interest. Where it came, where it is gone, is all, you know, all this politicians to bicker about, but reality is it is there, right? So can you go to next slide, please? Yeah, so these are the verticals that I talked about. And good thing that we did in that week itself, we decided that we will not do anything where a corporate partner is not there. So CSR is not doing any project where industry is not in partnership. And the right side, you see the industry which is working with us. Reliance Industries working, Tata Sons, Intel, TCS, Cipla, Sun Pharma, Arvindo, Cadila, Bharat Biotech, Syngene, BAIL, BL. And this list goes on. This is almost 15 days old slide. So all things that we are doing, because we very quickly, I realized, and I told everybody that without industry, you will only keep making noises. Okay, you will not move an inch in the real space. This is very important. And luckily these industry they came as a partner. And also some of the large industries came as a large corporate social responsibility fund. Like Tata Sons has given close to 3 to 4 crore rupees to us as a CSR responsibility. Reliance, all these companies have given money from CSR fund. At the same time, they have in-licensed technology. I will briefly talk about it. So this is something very important, the way we took approach and the rest of the people. Can you go to next, please? Yeah, so next. Yeah, so here I think this is covered. Briefly, I will tell you that so far, this is a little old slide. We have done genome sequences of 500 viruses, and those have been deposited to international repository. And from those viruses, we are learning a little bit about the mutations, but the most importantly, what we are doing, using those viruses, we have now set up the varicella in vitro cultures in 3 of CSIR laboratories, where we can test substances for antiviral activities. We are in the process of setting up animal model using those virus resources. And since we have industry partners, so we are trying to set up a public-private partnership where this in vitro, phenotypic, cell-based animal model, all this can be done in a private environment, and it will be provided to everybody as a service mode, rather than getting done things in a government laboratory at its own dimension. So that is how we are working with a Bangalore-based company to create a public-private partnership for this resource. Next slide, please. Yeah. So this is I think I briefly mentioned about this Tata Sons, and because of our effort, Tata has decided to create a new company called Tata Health. You will hear very soon the launching of this company that will focus on based on our diagnostic kit that we have given them, and that work is happening. Next, please. Next slide. Can I change slide from here? Yeah. So the second part was that we decided in the month of February that, if you remember, there were a lot of shortage of testing labs in the country. So 14 CSIR labs are now day-to-day testing. We have tested close to 1.5 lakh sample in last 1 month, 1.5 months. Lab at Hyderabad, Chandigarh, Jammu, my own lab. We have tested close to 20,000 samples. So what we are doing, we are working with the medical colleges and government system, respective state government. And since many of us have BSL-3 facility, BSL-2 facility, and we are offering those facility and providing sampling. And this number is old. This number should be 1.5 lakhs, the 40,000 that you are showing, because this is the 15 days old slide. And also what we are doing, we are training people to do testing, RT-PCR testing. So we have trained close to 250 people now, young people across the country, who can go to private lab, government lab, and do the RT-PCR testing using standard protocol. Next, please. Next slide. Yeah. So this is one technology that we have developed. Our lab, IGIB in Delhi, has developed. This is using a CRISPR-based simpler kit to detect the virus, as you see here. And this technology has been licensed to Tata Sons, and they have done testing in there, and they have taken this basically for the purpose of that they want to screen all of their employees in Tata Group. That is the idea of using this simple kit, 30-minute test. you can test your positivity and negativity. And this currently has been tested on, as a prototype, we have done the hundreds of samples we have done, and we are hoping to get any day the ICMR approval, and the production facility already has been created by Tata's, separate company has been created, and very soon you will have this point of care diagnostic kit available to the country. So this is one of the major highlight of CSIR contribution in this effort. Next, please. Next slide. Yeah, this is the second technology which we have done with the company, Biocon company called Syngene. This is very interesting platform where we are using barcoding and sequencing together. And we have a platform where we can test 50 to 70,000 samples per cycle. So for example, if there is a hotspot area where a large number of people are infected, you can take this, set up this platform, and you can do this test, and you can 50 to 60,000 sample together. And now this is actually a prototype has been developed by Syngene with our lab in Hyderabad, CCMP. And hopefully, I am hoping within a month, this will get approval from the regulatory system so that we can pan out in an area where the hotspot of the country, or for example, companies can take these systems and then screen their own people, to prevent sort of a community spread. Next, please, next slide. Yeah, this is the diagnostic kit my own lab has developed. This is a RT lamp. This is a very simple technique where you don't use RT-PCR machine. So as simple as that, you just simply need a heat block, 60-degree temperature, and you use 4 PCR primer. And this we have out-licensed to Reliance Industries, and Reliance Industry has set up the cost at 99 rupees. Mr. Mukesh Ambani himself is supervising this project, and he has set up that he will provide this kit at 99 rupees per test. And it is 30 minutes you can test whether you are positive or negative. This is also prototype has been developed. Reliance is working with its own industry system, and we are waiting for ICMR approval, which should come maybe a week's time or 10 days time, it should come. So these are the 3 very important technology that CSIR has developed. These are all our own patents, but we are making sure that we are not making these technology, giving these technology on exclusive basis to any industry. So other industry is welcome to come and take these technology, because these are developed by taxpayers' money, so it cannot be given to one industry on an exclusive basis. Next slide. Now this is the third vertical, which is drugs and repurposing of drug. Next, please. Yeah, so what we decided at beginning that, obviously it is not possible to develop new drug in that short period of time. So we selected 21 drugs which are potential antiviral substances or are in clinical trial for any viruses, particularly positive RNA viruses. And those 21 drugs we distributed about 5 or 6 chemistry-based labs: Hyderabad, Pune, Jammu, Lucknow, Bhavnagar, and we developed the synthetic method at least at hundreds of gram scale in the lab. So for example, remdesivir, before remdesivir was launched, IICT Hyderabad had remdesivir synthesis. My lab also has a remdesivir synthesis, favipiravir, unifiravir, Arbidol, all other drugs. So 21 drugs we have developed synthesis available to us. And then we started working with partner. For example, this is one for the favipiravir. Our partner is Cipla. Together with Cipla, we went to drug controller. Cipla conducted a trial, and Glenmark also conducted a trial, and Cipla got approval to market it. And Cipla is now selling it actually at 1/3 of the cost. That's what their target is, that they will provide favipiravir at 1/3 of the cost which Glenmark and other companies are providing. So I think that's the advantage that we are doing with the bringing competition into the system. Next slide. Yeah. So this is the remdesivir, and because of the intervention of CSIR, we developed a cost-effective method, and demonstration for pharma companies were run by IIIM and IICT both. And this technology has been out-licensed to Mylan, Laxai Life Sciences, and Cipla. So anybody who's coming, and we are helping them, giving this technology, and these are the people who have license from the Gilead, the originator Gilead, and they are producing it. So I hope the cost of remdesivir also will come down as these new processes in India start producing at large scale. Next, please. So these are the some of the top drug that we have identified, and we are putting in the pipeline. That first one is already in the phase 2 trial with CDRI Lucknow and a company. This is Arbidol. The niclosamide, we are working with the King's College London and a company, and very soon a phase 2 trial will be started. In fact, this application is now being prepared. And then we have the 4 combination trial, which is going to start. In fact, this week we are hoping to get DCGI approval, where we are testing. So my approach has been that, only antiviral will not help, only immune system directed things will not help. So we created innovative combinations to deal with the situation. So the 3 combinations that we are going to start trial is favipiravir and colchicine, Arbidol and colchicine, and nifomystat and 5-aminolevulinic acid. These are the 3 new combinations which CSIR is working with industry. Fortunately, we got a industry partner, and we are putting 50% money. And this, in fact, today or tomorrow, we are hoping to get approval, and then this trial will start. Next slide. And of course, this is the CCMB, the antiviral test, that system that we have developed, inactivated virus for the vaccine. So industry partners are written here, which are working with us in terms of the Bharat Biotech, VNS Biotech, Esteem Research. So many companies are working with us in this. Next slide. Yeah. So this is the collaboration. Very early on, I must thank Dr. Khamar, who is sitting here, that we very quickly, I think in the month of, January or February itself, we came together, analyzed this, that this is something sitting on in our hand. This is already approved for gram-negative sepsis, the Sepsimed, the MW product. And then we quickly created a project, and then very fast we went to DCGI, and we got 3 approval, 1 phase 2 approval, 2 phase 3 approval, which are written here. And I'm hoping that, we are almost at the end of the phase 2 approval, which is for serious patient. And we will in the process of analyzing this data, and very soon we will go back to, and in this effort, what we decided was to create a benchmark that these trials are done in the top hospital of the country. AIIMS Delhi is involved, AIIMS Bhopal is involved, and PGI Chandigarh is involved. Now 3 more AIIMS are joining, Nagpur, Bhubaneswar, and Raipur. So I think this is the kind of a partnership that has happened and created a benchmark for us. And I think this is the one direction I'm more hopeful to deal with this disease, because rather than going for the vaccines and looking for a specific activity and the problem that vaccines have, this is another way of dealing with immunotherapy. It's called basically immunotherapeutic approach to the virus, is to basically kickstart the Th1 protective response of the human body, which itself should deal with this, and of course associated IL-6 related damage that is happening. So this is something we are all looking forward to, that this data gets completed and we get into the larger trials. Next, please. Yeah, this is something very historic, I must tell you, that this I take very pride, and I was directly involved in this. This is, as of in 2015, India created a new type of drug category under DCGI called phytopharmaceutical drug, okay? And this is based on what we call US botanical drug, which was created in 2005. This is the new dawn in drug discovery, other than a small molecule and biologicals. Here, basically, you have a plant product. You do not have to go to the single molecule. You can use the advantage of, because many of these plant products work synergistically and additively. And this is the long-term demand that we should be allowed to develop these drugs, provided that the CMC and GMP all is followed. So this partnership, actually, we started 5 years ago with Sun Pharma and ICGV, and it started working on dengue virus. This is a material, and IIIM Jammu did almost all IND enabling studies was done, and we had fortunately completed phase 1 trial for dengue. And then, dengue virus is also positive RNA virus. And although the mechanism of entry of dengue virus and this virus, coronavirus, are different, but once inside the cell, the endocytosis, going to ribosome, N-link protein processing, everything is same. So quickly we tested this in outside the country for coronavirus. And then we went to drug controller. Since phase 1 data was already there, we got phase 2 data. This is also happening in 8 hospitals right now in India, all over India. And we have almost completed 80% of the patient recruitment, and maybe in a couple of weeks' time, we will have data. I think this is a discovery made in India, developed in India, and first phytopharmaceutical of India. I think this is the dream of Ramnath Chopra in 1930. And that is how we created this institute. So I take special pride in this, that I was associated with this, bringing first phytopharmaceutical of India into clinical trial. So I'm hoping that, you know, this will also come into picture. Next slide. Yeah, so we are doing these also. We then partner with the IIIM ministry to help them, and these trials are happening in terms of the additive trial, whether some of these very important plants, particularly my hope is on Glycyrrhiza glabra and Withania somnifera. And again, indirectly, Cadila is involved in helping us conducting these trials all over India. So you will hear about these trials also. Next. This trial is happening on you must be hearing about plasma therapy, conversion plasma. So we are doing, we don't go by, somebody said, you know, plasma therapy is working. So we decided that we must conduct trial. So our lab in Calcutta, IICB, medical colleges and other, this trial is happening, and DCGI has approved this trial, and this trial also going on for validation of plasma therapy. Next. Hospital assisted devices, next please. Here we have made several interventions. The most important intervention I will tell you is this non-invasive supplementary ventilator. This is a very portable device. For example, if you are in home and your oxygen level goes down below 94% and you are suddenly panicking, because nobody wants to go to hospital these days. Nobody wants to go to hospital. The hospitals are like going to death, right? Getting infections, dying there because of lack of facility. And so we developed this device. In fact, I'm very pleased to say yesterday it got regulatory approval, and Hyderabad-based hospital placed the first order of 25 ventilators. Very simple device. It gives you oxygen within 2 or 3 infusions. Your oxygen level goes back to 98% to 100%. So, and in that process, your lung have capacity to go back, and you know, most of the damage can be repaired. So it's very interesting. CSIR and a lot of companies are involved in this who will sell this. Oxygen enrichment unit, where we can convert to 60 to 70% oxygen from air. We can use membrane filter. And next slide, please. Next slide. Yeah. So here I will not take too much time. I'll only talk about our lab in Bangalore, National Aerospace Laboratory, which has nothing to do with the disease. They came forward in 3 months ago that they will manufacture PPE for hospitals, doctors and nurses. Because in the space area, they use these very high sophisticated gowns and to protect these pilots and other people. So they had technology. And today I'm pleased to say that with this technology, one company in Bangalore is making 50,000 ventilators every week. And their capacity is going to be 1 lakh per week. So I think this is we take special pride in getting that this technology came from CSIR, and it's very cheap, and it is being bought by all hospitals, private sector, everybody through the HLL system of the government of India bidding system. Next, please. Of course, there are a lot of things I don't have time, so I'm not telling you about everything. Next. Yeah, so these are some of the, now we are working on a project and we are discussing today. Can you go to next, please? Yeah, so we are working now these makeshift hospitals. So we are developing now, we have a technology where within a week, we can set up a hospital where 100 to 200 people can be managed. At least the mild to severe patient, mild to moderate patient can be managed in this. So I think we are, L&T is our partner, Larsen & Toubro and Tata project. Both of them are coming together with us, and this technology will very soon we will launch. Next, please. Next. Yeah, next. This is the supply chain. So basically here what we have developed online portal and apps where we can follow the key starting material of particular drug, which is coming from abroad, which is coming from India, and this database we are generating and providing to industry partners. and ourselves, our lab. So I think this will be the first of a kind effort in India where this kind of crisis happens, because two lessons we have learned, that over last 15 years, we virtually outsource all the drug manufacturing to outside India. And we became people who will make tablet, capsules, and injections. So we became basically, from the manufacturer of drug, we became formulator of the drug. This is the harsh reality. And there is a reason for that, business reason. And I think government is now realizing that what kind of horrible mistakes they have done not promoting API manufacturing in the country. So we are discussing now, a new policy has been announced by Department of Pharmaceuticals in consultation with us. 6,000 crore they have allocated, 103 drugs they have identified, and particularly drugs which are produced by fermentation. You know, drugs like rifampicin, and rifamycin, now we are importing from outside for tuberculosis. That's we used to produce in Pune facility, and even I think Kerala used to produce or something. So something like that. This is useful. Next, please. Yeah, this app, very interesting app that we have created for farmers to get connected to industry. So farmer knows which industry need these products, industry need which product is lying where. So this is, I think, very large use, more than, I think, this 35,000 is very old figure now, more than 1 lakh people are now connected to this app. Next. Next slide, please. Yeah. So these are the things, this slide I made especially for all of you. What hard lesson we have learned during pandemic. And this, we must ponder over it. If we don't ponder it, we will have bigger problem in our head. First is that pharmaceutical manufacturing base of India has shrunk to alarming level and shifted to China, Southeast Asia, and CIS countries. So two things have happened. Medicines may not remain affordable for Indian population, which was the dream of whole setting up pharma base. And I think that was a dream of founder of Cadila Pharmaceutical, which I had a privilege to meeting a couple of times. I think I personally came to Ahmedabad and met, and he was actually associated with the research council of my institute for many, many years. So he was like a guardian to us. And I think that dream is in the danger. The second dream, jobs. Good synthetic organic chemistry people don't have job because manufacturing base has shifted. So Wuhan has more Indian organic chemistry graduate working than in India. So I think all the synthetic chemistry ability which people like Dr. Yadav built in this country is in serious danger. I'm warning it. This is a problem. Okay, this slide I made is specifically here. Second is medical devices diagnosis sector. We are totally dependent for outside country. Every component, every IC, every piece, once you start assembling these, data realizer, everything's coming from outside. Even a capacitor is coming from outside. So this is all imported. And we are sleeping nicely that, we are, manufacturing base of the world. I think we have to get out of that sleep. See the writing on the wall. This is the serious problem. So we are now in discussion with Niti Aayog to have the policy to support a startup and bigger company to get into diagnostic space. The third, the most important, I'm telling you all this story that this drug we are repurposing, this vaccine we are repurposing, except Sepsivac and phytopharmaceutical, which are Indian innovator product. Everything is from outside the country. We have no contribution in remdesivir or favipiravir because these are discovered. So I think the discovery base of this country has close to come to zero, almost zero. Whether a small molecule, we have become imitator of the outside world. Somebody launches antibody, we start making in our country. When are we going to discover our own antibodies? After 70 years, having such a robust power, manufacturing base, everything. So I think there is a message in this that we cannot keep on copying and hoping that Western world will keep discovering drugs and vaccines for us. We have to do, now we are adult country, we should be able to do this. And for that, there are two problems. There is no translational expertise. You will have a lot of professors will give you a lecture here. But if you go in the lab, there is zero translation facility. If you have a molecule and if you have a good IC50 and engagement with target, they will say, I have discovered a drug. They even don't understand what it takes to develop and discover a drug. The entire ADME, PK, tox, IND is missing. And only few companies may have these capabilities. Rest in public domain, it is totally missing. No investment has gone to this. People don't appreciate this, that, you know, just having biological target engagement doesn't make you a drug. It's a whole journey that happens. So that neither the expertise is there, nor infrastructure there. And that is what is happening, that we are into such a difficult situation. So this virus is telling something very important, that without investing in innovation and discovery, rest of the industry is basically operating at 5 to 8% margin. How long you will operate? We have to go to the heart where the margins are 100 to 100%. Look at the profit Remdesivir Gilead must be making. You can't even imagine the amount of profit they will be making, because they are the owner of the intellectual property of discovering a drug. So I think this is something is important. And these are the one which requires immediate attention from the public system and the private system both. That without innovation and discovery, although it takes time, I understand a lot of people are negative, you know, all this negativity is all right, but that's the only route to create wealth and stay relevant in terms of these crises. So I think this is my last slide. I will thank you for inviting me here. And I have given perspective how CSIR has contributed and will continue to contribute. You will keep on hearing more things from us. And I'm very pleased to be associated with this particular discussion. Thank you very much.
Thank you so much, sir. Actually, you updated us about the strategies and measures which are taken by CSR to contain COVID-19 with the help of government and with the help of industry, PPP model, all that thing. That was very informative. In fact, we have a strategy at the front also, where soldiers are fighting, and they have a strategy, similar strategy the CSR is doing, you know, to contain this COVID-19, which has scared everyone globally, in fact. So that was wonderful information, sir. And the measures taken are really appreciable, especially when you raise the concern that it's a high time now to government to think on ROI, that is return on investment, you know, which they have been raising, that what is the ROI on investing on science and technology. This was actually wonderful. And it's high time that everyone has to open their eyes and seriously think that how much investment is required on science and technology, especially when we are witnessing this. And it is not happening at the country level, it is happening at cross-country levels. So that is high time, and we are simply dependent on imported things, you know, for so many things. This was wonderful, sir, and it has sensitized us and the seriousness of COVID-19 also. And now, if you permit, we'll have a couple of questions if time permits. Can we start, sir? Yeah, and there is one question which has been asked. Can you suggest a sequence of medication for a home quarantine COVID patient?
Yeah, so home quarantine COVID patient, now I think, of course, it's all should be under medical supervision, but there are options now. So now I think after a lot of controversy and a lot of this, I think in last 3 months, thanks to science, we are in a much better place. Particularly the breakthrough that came from Oxford University recovery trial, the data on dexamethasone, I think that has made all that difference. So according to me, the only disruptive change that has happened in treatment regime so far is dexamethasone and prednisone. So if you talk to doctors, all over the world, doctors are saving many, many lives because of this intervention. And and other is this, despite all the criticism, hydroxychloroquine has a window of opportunity for people when they are very early phase, when virus is just entering. And you know, hydroxychloroquine works with a very interesting mechanism that by modulating pH gradient, when virus is trying to get into from endocyte to endosomes to lysosome to then going to the ribosomes, right? So that's where there's a short window about that. And fortunately, the recovery trial on dexamethasone, they did one more recovery trial on hydroxychloroquine, which is published yesterday in I think, New England Journal of Medicine. And one thing they have conclusively said that on 600 patient trial data, that all the concern that were raised about hydroxychloroquine safety, absolutely there were no concern of the safety of hydroxychloroquine. And India is one of the countries which has used this drug and saved millions of lives in almost 40 to 50 years. Dr. Yadav will remember, and I will remember when we were kids, you know, when you used to go to school, Morning we were given one tablet of hydroxychloroquine every day because there was a time of malaria epidemic in Uttar Pradesh. So people like us who are sitting with you, we are alive. At least I am alive because of hydroxychloroquine. I'm telling you, otherwise I would have died in childhood because of the malaria, right? So there are this, and then there are, of course, the molecule like colchicine, 6,000 patient trial data. And, JAMA has published a study. It is really helping in the early phase of basically maintaining the pro-inflammatory responses of inflammasome and IL-6. So there are a couple of medication which are available, which I'm sure doctors will prescribe. I cannot tell you exactly that you take this. That's unethical for me to tell you, but we are in a much better place than 1 month ago. I can tell you. Then the one most disruptive data came from Oxford University trial, because that was such a large trial, 156 hospitals involved, 6,000 patients, and they have put that data in public domain. And I think that has made all the difference. That I think, first, I'm not too sure about remdesivir and favipiravir, how good they are. Doctors are not really as yet convinced about it, but Dexa and prednisolone certainly are helping, and hydroxychloroquine is that is where I'm hopeful for Sepsivac. If the Sepsivac comes, and I think if we can really protect people from going into serious situation, I think that will be one intervention where we can really, and I think the data that came from BCG trial data, which is hinting towards, I think, the hypothesis that Dr. Kamar and I have been exchanging papers, and I think that hypothesis is being slowly being validated, that non-specific management of innate response is a better way to deal with, rather than, going all the way to, in fact, yesterday, New England Journal of Medicine has published a very beautiful commentary on this topic, Dr. Kamar, and I will share with you that, saying the same thing, that's the way we have to look at it. Yeah.
Thank you, doctor. The other question is, patients, when you talked about Sepsivac, it is related to that. Patients who are critically ill, in spite of giving steroids and other medicines, can we try Sepsivac at that time?
Yeah, we can, because if we have to deal with a sort of the cytokine, what we call CRS syndrome that happens, a lot of these, particularly the IL-6 and others. I mean, particularly the trial that is happening actually for the seriously ill patient, the phase 2 trial that we are doing is on those kind of patients. So I think if they are we can slow down their movement from the less critical stage to higher critical stage, and use other interventions. So I think this Sepsivac can provide that window for doctors to intervene in terms of patient progressing faster. So that I think that's that window available to them. And I think I can see the combination of Dexa and this will make a whole lot of difference. Because Dexa will actually will work through a different mechanism, and also it will provide some anabolic, because basically Dexa and prednisolone are for basically glucocorticoid and mineralocorticoid balance. So basically Dexa is more towards glucocorticoid. This is about more towards mineralocorticoid. So I think that is also important, providing body to more metabolic strength for body to deal with this. It's basically tilting the balance. Body is trying to go into overdrive to protect you. Now, if you have to slow down that overdrive on one hand and slow down this virus replication process, but when you are in second week of infection, you are not dealing with virus, you are dealing with immune system. As simple as that. So if you can protect yourself from your own immune system, I think you can protect life.
I think Dr. Bakresh would like to add on that.
Dr. Bakresh want to answer that question.
Sir, please unmute yourself. Sir, please unmute yourself. Sir, please.
Okay, Dr. Yadav, you wanted to say something, then I'll add whatever Dr. Ram Vishwakarma said. Whatever I know about Sepsivac, I will add to it. Go ahead, Dr. Yadav.
No, actually, I thought you were raising hand to say something.
Okay, so Sepsivac is already available in the market, and essentially it is approved for gram-negative sepsis, where underlying conditions are identical to what we see in COVID-19. Based on this, we propose a trial, and trial is underway, But since the product is available, doctors have started using it. So nobody need to wait. And actually, Karnataka government has put it in that protocol that any patient who is seriously sick or critical needs oxygen, they need to get Sepsivac. So currently, there are no guidelines for rest of the country, but I believe now Surat as a city has produced their own guideline, and that guideline also includes Sepsivac. And large number of doctors have started using it, and they are saying it saves life and it makes recovery very fast. So this is what is possible, but the trial data, as Dr. Vishwakarma said, is yet to come, but it works. I mean, so there are two types of therapy. Either you work on the organism or you work on the host. So Sepsivac works on the host, and it improves host capability to deal with whatever eventuality organism is doing. So almost everybody who gets infected does not get infection. This is known for all organisms which are mainly intracellular. And the reason we don't get is our body is capable of destroying it. If it enters, our body is capable of taking care of it. And when it works, our body tries to reverse the situation in absence of any therapy. So what body does, the Sepsivac tries to help body do it better. Yeah. So whatever inflammatory immune response or a cytokine storm is body's effort to get rid of an organism, but it is inadequate and it has gone haywire. And because it has gone haywire, because it is inadequate, we get a condition where people die. So either the host dies or an organism dies. So in this condition, when the host is likely to die, if you enhance the efficacy of the host, then host will survive and organism will die. And that's what Sepsivac is likely to do. And this has been demonstrated in gram-negative infection, which is again an intracellular organism and does the same thing like what COVID-19 does. It induces immune response. That immune response makes the people critical, vulnerable. They put them into ICU. And the whole trial was done in the patients who were under the ICU. And in that trial, what we observed was that if there are 100 people likely to die because of gram-negative sepsis, we use Sepsivac with the standard of care, 45 will die, 55 will survive. And based on that, it is approved and available in this country. Yeah.
Thank you, sir, for adding on it. If you permit, we can take another question which is frequently popped up, and that is how is the herd immunity, if India reaches to, can help to combat or curb against COVID-19?
Yeah, so I think that is the part of the basic biological processes, because this is not first time that we have this virus. These kind of viruses, if you read the history, they come every 200 years. Every 200 years, these kind of very, very infectious virus come along and they disappear. And they disappear through their continuous interaction with the immune system. And I think immune system is basically created for ourselves to survive in a microbial environment. That's how and I also, let me tell you this in a poetic sense, that this is our most ancient ancestor who is visiting us. The life on Earth is started with the RNA virus. That is the first life form that evolved from inorganic world. So this is as old as the life on Earth. So these viruses have been interacting with life forms, and herd immunity, when we talk about, certain 40 to 50% people, but there is a risk. That is where we need a strategy. We have to protect people with comorbidities, people with susceptibilities, and it will go to certain number of people. And when it moves from human to human, it will get weakened and weakened. That is how the immune principle works. So eventually it will have to be dealt by that mechanism also. But we have to be smart so we don't lose more people. We protect our vulnerable people, comorbidity. And that is where I think the next opportunities for pharma industry is that we need now, having done repurposing and all that, now we need to look at the combination of medication, where how do we therapeutically protect comorbidity people? how I protect a person who has type 2 diabetes from COVID infection. Okay, how do I protect somebody who has got, say, immunocompromised state from infection? So these are the next generation challenges, and we have started doing this thought process. Yesterday, fortunately, we had a discussion with a very large US company on this aspect. So we are moving into scientifically in that direction also.
Thank you, sir. And there is one another controversy going on in the news these days regarding the N95 mask, how far they are safe. You know, there is a lot of hue and cry that whether with the socket or without that, you know, whether it is helpful or not, or whether it is more harmful. So whether they are detrimental in spreading COVID-19. So can you comment on this?
Yeah, so anybody who's saying that any mask is harmful is lying to you. Don't listen to that. Mask is the only protection right now. I will tell you very upfront. You are only protected by mask, and particularly after this discovery came from a Nobel laureate's Molina's lab 10 days ago, that what is happening in particularly humid environment like India, the 20 micron particle which is there in the air is dehydrated and converting into 4 micron droplet. And that droplet stays for several days in the air. Let me tell you this. These are the two very important paper published in very prestigious journal, Proceeding National Academy of Sciences. So that is why now WHO has revised its guideline. India has revised its guideline. The best protection that we have, even if you're nothing, you know, even cloth mask is good. We need to protect even when we are talking to each other. We are making presentation or having a meeting. I think any mask is better. Of course, N95 is ultimate. If a good quality masks are available, that's very good. But mask wearing, whether you are for yourself or outside people, I think mask is the only protection right now. Those countries which have not followed this, see the graph. Europeans have followed, their graphs have gone down. Americans have not followed, their graphs are going down. So I think this is a lesson we can learn, that wearing mask is an absolute must wherever you are. So don't think I'm alone walking in my garden, you know, I can remove it. No, because if somebody has walked before 5 minutes you, and if he has done this, then particle is sort of aerosol is suspending for 2-3 hours, you are going to get it. So that's very critical. This is a new dimension that has come after the Molina's study published. I can share that paper with all of you. It's hardcore data, very valid data, and that is why then people then wrote to WHO that please revise your guideline. The earlier guidelines is that you are alone walking, don't wear mask. Now that has to change. You never know who has come there before you, or even you. So I think it's important to wearing mask. And all these controversies are basically for, you know, people that spread it because of superstition and all kinds of things. I think. Yeah.
Thank you, sir, for removing this confusion. Now, if the panelist permits, then we can take, because questions are many and time is ticking. So as you say, sir. Bakri,
Okay.
sir, as you say, sir.
Thank you, Namrata. Thank Dr. Ram. You have very nicely explained not only SEPSIVAC, but whole area of what CSIR is doing, and I'm sure it is very informative. Now, I request Dr. Kumar. He is the Executive Director of R&D Cadila, and he's the one who has worked on SEPSIVAC. And, you know, he had studied abroad in India, a lot of experience he has, and which is not mentioned in his thing. He is one of the best eye surgeons in Hyderabad, Hyderabad. Best eye surgeon in Ahmedabad, and he's so dedicated that early morning he does surgery, and then in the morning again he will be in the lab, late night he will do again in the time. So he's one of the most dedicated scientists, and he will give some concluding remark. And if you have any question, you can ask. Actually, somebody had asked one question, Dr. Kumar.
Okay.
Somebody has asked question. The question was like this. Can I have the phone number of a Kadla doctor dealing with SEPSIVAC? It is urgent, please.
So you
He's Dr. Rajiv, his name.
yeah. So give your number and somebody will approach you.
Okay.
Okay. So So anybody who needs a sepsivac, please give your number or ask your chemist to get in touch with Cadila guys and they will provide you. There is no shortage of sepsivac currently. And there will not be shortage of sepsivac because it is only indigenously produced. We are not dependent on any ingredient which comes from outside. So don't bother about the shortage. It will be made available wherever you want it. So just give your number and somebody will contact you.
He had given already the number.
Yeah, okay, I have not seen it.
89855. Can you note down? He has given already the number.
Yeah, so we will ask somebody to get in touch with you. Okay?
Okay, okay. Okay.
Yeah, I now request Dr. Kamar to have his concluding remarks on this webinar.
So, I mean, as Dr. Arvind Sahu and Dr. Ram Vishwakarma said, COVID-19 is something which came out of blue. Nobody was aware and nobody knew what to do. But as time passes, everybody puts in their efforts to know more and more about it. And India as a country is not lagging behind. You heard CSIR efforts, which is on the forefront of this fight against COVID from India. And we know much more about COVID-19 than what we knew. But unfortunately, one thing which is common, which persists, will persist, and that is we need to maintain discipline, hygiene, and distancing. There is no shortcut to that. If you want to control coronavirus or COVID-19 epidemic spreading, you need to maintain this and you need to protect yourself from getting infected. As Dr. Ram said, you need to wear masks and you need to maintain cleanliness whenever you touch somebody or whenever you get in touch with somebody. So unless you do this, you can't be protected. And if we as an individual do this, we can make our communities, our societies, our cities, our country safe from COVID-19. Vaccine will come, but it will take its own time, and no vaccine will be 100% protective, not expected, and there will be a lot of challenge. So while we wait for a vaccine, which is likely to happen in near future, but we should do whatever we can do as an individual to protect ourselves, our families, and our societies. Having said that, I mean, there are a lot of things which are happening. So as Dr. Ram Vishwakarma said, CSIR is working on all fronts. As our Prime Minister said, we need to be Atmanirbhar in this area, and CSIR is on the forefront to make our country Atmanirbhar to take care of not only this epidemic, all future epidemic. And we are moving towards having our own innovative system geared up. And in that respect, Indrashil University has an utter innovation mission just to groom younger individuals who has some bright ideas to take it forward and get their all guidance under one roof, all infrastructure under one roof. So that's utter innovation mission where, and this utter innovation missions are across the country established with the help of Government of India and universities. People with the bright ideas can go there and make their ideas fruitful or reach to the next level. On terms of the host side, we say that SAPSIVAC is one example, and in future there can be many more examples as we move forward. And Dr. Ram is very fond of host-directed therapies, though he himself is chemist, synthetic chemist who works on a small molecule, but he has changed his way from a small molecules to host-directed therapies. And I'm sure we will have a small molecules which work on the host-directed therapies. With this, I'll conclude this session.
Thank you so much, sir. Now it's time to wrap up and tender vote of thanks. At the same time, I see that the webinar was attended by our academic council member, Dr. Acharya, also Damodar Acharya sir, and many more from Andhra Pradesh and Dehradun, and the enthusiasm which was shown by the attendees and participants is actually to be thanked here. Now officially on behalf of Indrashil University, I convey my earnest thanks to our experts today, Dr. Arvind Sahu, who has left for his preoccupied engagement, Dr. Ram Vishwakarma, who is with us, Dr. Bakulesh Kumar and Dr. Yadav, for accepting the invite and sharing their thoughts on the measures, research, and strategies which are carried out by CSR with the help of government, industry, and the individuals also are involved here who are dedicated, like Dr. Ram Vishwakarma is there individually. He's taking, his interest to curb this global pandemic. I'm sure the participants might have perceived clear understanding of the efforts being made by the CSIR and various research agencies to contain and mitigate the scary virus that has taken the world to a storm today. In fact, it has shown us today with all the measures which are taken, especially with the CSR, what Sir has, Dr. Ram Vishwakarma has said, that multi-layer vertical approach has been taken by CSR since, January. So these are all, showing us a ray of light at the end of the tunnel that India will very soon overcome from this difficult and dark time. In fact, the entire world, I wish. I extend thanks to all the participants once again for their presence and interest shown for this webinar. The entire IU team, admission team, AICIC department and management are also acknowledged with modesty for their efforts and incredible arrangement for this. I wish everyone to be safe, stay healthy, stay alert. Jai Hind.
Okay. Thank you.